Welcome back to the lecture series in bioenergy. So, the first terminology which I am introducing here is photosystem 1, photosystem 1 or sorry photosystem 2, and which is out here a colony of there is another colony of chlorophyll molecules which is sitting out here, which is called photosystem 1. Okay. Now, what exactly happens is in both the cases the light falls, both in photosystem 1 as well as photosystem 2. Okay. Now, in both the places an electron is being ejected out, here also, here also. Okay. Now, the electron which is generated in photosystem 1 is involved in generates a reducing power because it is an electron. So, it can reduce, generates reducing power in the form of NADPH. In the form of NADPH. Okay. Now, having said this, so now photosystem 1 has ejected an electron because of the light. So, it means there is a chlorophyll molecule which is oxidized. So, that has to be brought back to the ground state. Just follow my previous slide, I showed you. One second, here. So, this has to be brought back to its ground state. So, just now getting back to the slide. So, here the electron which is ejected, this has to be brought back. The electron for the photosystem 1 is ferried from photosystem 2 through a series of mechanism which will be coming later. So, there is an electron transport which takes place along the membrane which we call as a thylakoid membrane. Now, I am drawing the membrane out here, where same time while light has fallen on both photosystem 1 and photosystem 2, the electrons have been ejected from the chlorophyll molecule in photosystem 1 as well as photosystem 2. The electron which is ejected from the photosystem 1 helps in forming the reducing power of NADPH, what we have talked about. If you see the slide, generates the reducing power in the form of NADPH. So, at this stage, one chlorophyll molecule or a population of chlorophyll molecule present at photosystem 1 needs to be reduced or brought back to their ground state because they are all oxidized. So, these electrons which are ejected at photosystem 1, say for example, these chlorophyll molecule CHL plus which are devoid of, are being brought down to their ground state by the electrons supplied from photosystem 2. Okay. So, these are the electron traveling from photosystem 2. Now, there is another situation. If the electron has been supplied by photosystem 2, so all the chlorophyll molecules which are present, now think of it, think of the situation out here, there are series of chlorophyll molecule which are now at a higher state. They all have lost electrons and they are all again, they are also oxidized. Now, who is going to bring them back into their ground state? This nature does with an amazing efficiency, and which is a challenge for mankind to understand. Just underneath photosystem 2. Now, I am just highlighting that zone. See, I am highlighting a zone, you see there. This is where there is a nano or atomic machine sitting there, which is called manganese cluster. This nano machine or atomic machine takes up the water molecule and it just strips out the water molecules and becomes a perennial source of electron, and as a byproduct it gives out oxygen. Making sense? So, this is that electron or the family of electron which is coming by ejected out by the water splitting are the ones which brings the chlorophyll molecules of photosystem 2 to their ground state. So, this is how. So, this is PS2 and here you have PS1. Okay. So, you see the whole thing is nothing but oxidation and reduction, oxidation and reduction. But end of the day you need one perennial electron donor. In this case, that electron donor is nothing but water, which is abundant on the floor of earth, and in that process it generates hydrogen. So, there are a lot of research and we will talk about this manganese cluster, we will in depth about it. There are a lot of research which are going on in the field of inorganic chemistry, where there are people who are trying to emulate the manganese cluster. If you could emulate the manganese cluster and we could split water, then we can generate hydrogen, and this hydrogen could be the source of infinite energy. But as of now, we haven't been really successful to match the efficiency with which nature is continuously breaking water into oxygen and being the fountainhead of electrons. So, this is that unique zone underneath photosystem 2. But then is this electron transfer which is happening from photosystem 2 to photosystem 1, what I have drawn out here, if you follow this thing, like I am just putting it in the pink now, this is this a straightforward process? Actually, it is not a straightforward process. This electron which is generated out here is, so there are series of proteins which are present out there, which are taking part in a phenomena called electron transport chain. So, electron transport chain. So, this electron transport chain, for those of you who have read about mitochondria and biology, will be knowing this similar electron transport chains are being seen in mitochondria too. Only difference between chloroplast and mitochondria is that the source of the high potential electron. In the case of mitochondria, the high potential electron source is the burning of the fuels, and in the case of photosynthesis, which takes place in chloroplast, the high energy electron source is the photoexcitation of the chlorophyll molecule. This is the most fundamental difference between that high energy electron which is generated in chloroplast as well as in mitochondria. Okay, coming back. So, now the electron transport chain. So, in the electron transport chain, the first molecule which takes place, and we will talk in later in depth about all of them, is the quinone molecules. So, there is something called a Q, I am just representing by Q. So, what is happening is that these proteins which are present in the electron transport chain or which are facilitating the electron transport are standing at different reduction potential. We will come later into that, what does that mean, the different reduction potential, different. So, what is happening is like a staircase. So, it is sitting here, it is accepting an electron, getting reduced, then throwing the electron to the next one. Now, this one is getting reduced, throwing the electron to the next one, and again coming back to the ground state. This one is getting reduced, throwing the electrons. So, it is something like, though I have drawn these proteins like this, but they are actually sitting something like this or something like this in a staircase model. And during this phase, this process, if you talk about the speed by which electron is getting transported in biological membranes, that speed is very less as compared to the electron transport in a conductors, what we talk about. But this slow transfer has its own flip side. This stepwise transfer through the different proteins which are present along the electron transport chain helps the membrane to maintain a proton motive force across it. We will come later into that, how that proton motive force is generated. So, what you see out here, coming back to the slide, across this membrane, what you see is something called a proton gradient, and this proton gradient is critical in generation of ATP or adenosine triphosphate, the other molecule. So, you have talked about one molecule which is NADPH, the other molecule what we talk about is called the ATP. So, let us summarize photosystem 1, photosystem 2, the two colonies of chlorophyll molecules present on the thylakoid membrane. Okay? Now, the light falls on both of them. Photosystem 1 and photosystem 2 not only are in space, they are sitting at different location, these two colonies, they are sitting at a different reduction potential. We will come later into that. So, in other words, their power or their energy to eject electron is different. So, not only they are located at different positions of the thylakoid membrane, the power with which they throws an electron out is different. The reduction potentials are different. Now, when photosystem 1 throws out one electron, it helps in creating the reducing force of NADPH, which further we will come to that, what is the use of that. Whereas, those photosystem 1 electrons or chlorophyll which are devoid of electrons are in an oxidized state. So, they have to be brought back to their ground state. They are being brought back to their ground state by supplying the electrons or transferring the electrons from photosystem 2 to photosystem 1. But while doing this process of transferring the electron from photosystem 2 to photosystem 1, it follows a cascade from one electron transport protein to another, to the third one, to the fourth one, likewise so on and so forth. The reason for following such a slow transport process is that during that process, it generates a proton gradient across the thylakoid membrane, and that proton gradient generates sufficient energy which is essential for synthesizing what we talked about here in the slide is the ATP molecule, which is adenosine triphosphate, the major currency for all the cells to function, adenosine triphosphate, ADP, AMP and ATP. Okay. So, now what this NADPH is doing? So, there are two more terminologies which I wish to introduce in this roadmap. So, roadmap is by far it's clear to you guys. So, you have photosystem 1, you have photosystem 2 and you have something called a network or protein wirings using different proteins sitting at different reduction potential, and underneath photosystem 2 you have what we call as the manganese cluster or water splitting cluster, which is the perennial source of electron. Now, we will add two more terminologies before we wind up the whole roadmap. Okay. So, this reaction could be divided also into two parts. One is called light reaction or light dependent reaction, and the other part is called dark reaction. what is the difference between the two. So, if you recall the first reaction, what we talked about? We talked about CO2 plus H2O forming CH2O plus oxygen, right? Now, this conversion of CO2 or this reduction reaction, what is happening between from CO2 to CH2O or to the carbohydrates, this is what we termed as, and this for this you need a series of electrons to take care of this in the form of the reduction potential. That reduction potential is supplied by what we talked about, the NADPH, and the reaction in which this whole thing takes place is called dark reaction or Kelvin cycle. Okay. So, the Kelvin cycle is essentially, so is basically what is happening is that CO2 is getting converted into, we will come in depth of all these things later. Just at this stage have an idea, phosphoglycerate, and all these things are happening within that organelle what we talked about as the chloroplast. So, within the chloroplast there are, and we will talk about this structure. So, you have this thylakoid membrane, but essentially this kind of reaction, the dark reaction is taking place outside it, which is also called the stroma of the chloroplast. On the stroma of the chloroplast, this set of reactions takes place, whereas the light reaction what you are observing is happening here, where you have photosystem 2, you have photosystem 1, which is generating an electron. This electron is the driving force for formation of NADPH, and whereas through electron transport chain, electron is traveling from photosystem 2 to photosystem 1 to bring back the photosystem 1 to its, from here to its ground state. Similarly, photosystem 2, once it goes up, it is being brought back to its ground state by the water splitting cluster. So, out here what you are seeing is, this is what is leading to the generation of oxygen, and second thing what is coming out of this is the electrons. Okay. So, if we have to summarize what all new terminologies today we in the roadmap we talked about photosystem 1, now I am circling all of them. We talked about photosystem 2, we talked about water splitting cluster, we talked about this part which is generation of ATP because of proton motive force across that membrane. So, if you remember in the beginning I was telling this, if you just go back to the very first slide, at such reaction center the energy of the excited electron is converted into a separation of charge, in essence light energy used to create a reducing potential. So, that is exactly what is happening, proton motive force which leads to the generation of ATP, is of course a part of the Kelvin cycle essentially out here, and on the other side this NADPH which is formed is running the Kelvin cycle, and this is where we talked about the whole biomass formation. So, if you kind of drag your imagination a little further, you will realize the efficiency with which light is being trapped by the chlorophyll pigment present in the chloroplast will decide how efficient the reaction will take place. Apart from it, mind it, this is not one standalone phenomena happening in the chloroplast. So, whenever such thing happens, there are other things which are also happening. Say for example, while the biomass formation is taking place, if there is an energy molecule synthesis, similarly there is another thing which is happening, which is energy molecule consumption. So, energy molecule consumption, which is also a process called respiration, okay, where plant or any other life form is using this process to generate energy for its livelihood. Apart from it, and this energy molecule synthesis, which is a light dependent process, unless otherwise we are talking about a hydrothermal vent, this leads to whole sort of photo damage. So, it is always a very trade off of three words: synthesis or anabolism or making something, respiration or a process of catabolism, breaking of the molecule, and the efficiency of the machinery, efficiency of the machine. And why this is so important, that efficiency of the machine has to be kept in mind, because of the simple reason that any of the atomic or molecular machine has a finite life. If there is excess damage, then definitely its life will shorten, and if there is too much solar radiation it is continuously exposed to, there will be more damage. So, how we balanced it out, and based on that, in the later half of this section we will talk about C3, C4 plants and how they vary. So, what you overall see, the biomass production. Now, if I had to pull everything together what we have talked about, the biomass production process, which is governed by the process of photosynthesis, and photosynthesis in itself is divided into light reaction and dark reaction. Dark reaction is the process where the biomass formation takes place, and the light reaction is the array of reactions which leads to the electron transfer through electron transport chains in the chloroplast by creating a proton motive force, which eventually make it happen, the whole process of biomass formation. So, with this brief overview, we will kind of moved on to this. Pretty much covered our first two lectures what we talked about in this section. In the next two lectures what we will be talking about is the whole process of how each one of these steps were discovered. Say for example, whenever if you see this reaction out there on the slide out here, if you see this reaction which is so how we discovered that. We will talk about chloroplast, structure of the chloroplast. Next segment is our chloroplast structure A. Then we will talk about the basic discoveries which leads to our understanding of. Then we will talk about reaction center and the discoveries which leads to the discovery of reaction centers. Then we will talk about photosystem 1, photosystem 2, and in between we will have few points here and there. And then we will talk about manganese cluster or water splitting. So, each one of these segment what we will be dealing with, and then from there we will talk about this biomass formation in terms of C3, C4 plants and evolving technologies. After that we will talk about evolving technologies for increasing chlorophyll and photosynthesis rate. And of course, eventually we will talk about, maybe another section will talk about the research towards artificial leaf. So, in summary, if we look at it, it is basically the process of trapping sunlight and converting it into energy rich molecule. So, I will close in here with this, and we will take up each part of it in our subsequent lectures. Thank you.